Thank you for introduction. Hello, everyone. And if any of our investors or collaborators are listening to this today, then thank you for your support. We at Exo aspire to profoundly impact the way cancers are treated. 2021 has been a very important year for us, and I will be happy to share with you what we have achieved. Founded 5 years ago, Exo is a preclinical stage company. Our R&D is located in one of Israel's biggest industrial parks. So our neighbors are companies that have been making headlines with their technological, clinical, and business achievements. Exo fully owns its IP, based on which we are developing first-in-class biologic targeting oncology indications of high unmet need. Our target are cancers with mutation in gene encoding p53 protein, and these comprise astounding 60% of cancers. I am proud to acknowledge our talented, experienced, and passionate team. Dr. Tendler Sr., who is now our chief scientist, dedicated decades to basic research in cell biology. He never imagined that it will prompt Dr. Tendler Jr., his son and my co-founding partner, the idea of this novel therapeutic approach. Our advisors are pioneers in their respective fields, and they combine outstanding academic careers with successful entrepreneurial record. And there are two more very special players on our team. The p53 protein is known as guardian of the genome due to its central role in cell response to DNA damage and stress. p53 has found to be mutated in approximately 60% of cancer cases. Moreover, cancers with p53 mutation are known to be more aggressive and therapy resistant. Well, while this has been known for 40 years, there is still no widely approved therapy targeting p53 mutated cancers. p53 is our therapeutic target. Unlike p53, exosomes, or more generally extracellular vesicles, have appeared on the therapeutic radar relatively recently. These small bodies are produced by all living cells. They carry active cargo, and they play an active role in intercellular communication. Exosomes are known to be non-toxic, non-immunogenic. They pass body barriers, allowing to drug previously undruggable targets. While today there is still no approved exosome-based therapy, many are on the way, and the field of exosome-based therapeutics is growing with CAGR of more than 20%. Exosomes serve as our delivery vehicle. Our exosomes originate from unique tissues where cancer occurrence is extremely rare, corneal epithelium. It was discovered that unlike any other, corneal epithelial cells contain at any given moment substantial amount of p53. Moreover, these cells secrete exosomes already loaded with p53, which in turn transfer them to surrounding cells. This creates a local mechanism of anti-cancer defense that evolved through hundreds of million years of evolution and is shared today by all vertebrae. So our core idea is to recover this vastly abandoned by our bodies mechanism and translate it into therapy. We harvest those exosomes, we purify them to ensure that there are no intact cells or other elements that would otherwise cause the body to react to them, and we administer them as therapy. The name of our company actually stands for Exosome Protein Therapeutic. We use native exosomes to deliver wild-type p53 to tumor cells. Use of exosomes ensures high therapeutic index and allows to pursue flexible administration paths. As there are no intact cells or DNA in our material, many of the safety concerns attributed to cell therapies are eliminated, and our therapy belongs to the emerging category of acellular biologics. Clinically, this translates to selective apoptosis of tumor cells and potential sensitization to conventional therapies. P53 has been a very attractive target, and we believe that with our approach, we're actually addressing pitfalls of previous P53 targeting therapies. Instead of human P53 gene, we use chicken P53 protein as an active agent. And instead of viral vectors, our delivery vehicle is exosome. Our in vitro results show P53 specific activity, where cell lines with P53 mutation react stronger than the non-mutated ones. And we also see no effect on non-tumor cells, which is a good first indication of the product safety. Observed cell dynamics, which I'm showing you in this time-lapse video, is consistent with cell cycle arrest here and the following apoptotic process, as opposed to just rapid cell death that would be caused by a cytotoxic agent. We have achieved some spectacular preclinical results working with immunocompetent mice. In the survival study on the left, we observed not just significant difference between the treatment and control arms, but actually some of the treated animals survived and died much later from natural causes. And throughout the treatment and later on, they actually had very good quality of life. There was also significant reduction in metastases following the treatment. Note that this effect was achieved with a single injection of the therapy. In the study shown on the right, we monitored tumor growth, and that was following 2 injections of the therapeutic. The difference is profound. Moreover, we followed other widely accepted markers. The histopathological analysis showed that there are other good things happening, namely reduced angiogenesis and increased apoptosis in the treatment group, and that is weak after the injections, demonstrating enduring effect of every dose. In neither of these studies, nor in a dedicated acute toxicity study that we actually performed even earlier, any toxicity signs were registered. So naturally, we're very pleased with these results. And armed down with these results, we're continuing with our program. Out of vast variety of cancers, we identified a short list of high impact indications based on the frequency of P53 mutations, degree of unmet need, and the opportunity to highlight unique advantages of our product. On top of this list is GBM, glioblastoma, the most frequent and the most lethal type of brain tumors, with the current 5-year survival rate of only 5%. There are many reasons for us to select glioblastoma as our first target indication. We want to make great impact on patient lives, and we also want to reach market fast, and we also want to make good business. So GBM is an area of acute unmet need. It is brutal, and patients are given today not a diagnosis, but a death sentence. No drug has been approved for glioblastoma in the whole last decade. We aspire to bring to those patients in need an effective and also safe drug without the devastating side effects of existing therapies. Pursuing the GBM indication will allow us to get organ drug status and a number of priority designations from the FDA. This will result in smaller and faster clinical trials and faster market access. The market size for glioblastoma alone is supposed to reach about $1.5 billion by 2027. This does not sound as a lot as compared to other mammoth-sized oncology markets. But just for a quick reference, the single FDA-approved therapy for glioblastoma during last decade a Novocure device, and it actually is an Israeli company as well. So the Novocure device only delays progression, yet market value of this company today is about 13 billion. Our GBM program is aimed at reaching clinical trials in GBM in 2 years and is targeting US market. This year, we have achieved important preclinical results. We generated dozens batches of material, and we started initial informal interaction with the FDA through Special Rapid Response Program. These days, we are raising bridge funding towards Series A next year, and we're expanding product development activities. We plan to reach interact consultation with the FDA early next year and to continue working closely with the agency throughout the development. Our work plan is built around a series of milestones directed at continuous de-risking of the program. Today, we're actively developing clinical partnerships, and we're also developing contacts with big pharma. As far as breadth of application claim is concerned, we have accumulated evidence in multiple cancer types beyond glioblastoma. Our vision is to expand to tissue-agnostic positioning through all-comers trials further on, and that will ensure access to the total addressable market of about 200 billion. Success of our program will to a large extent be determined by the success in developing production processes. We know that. They need to be robust and acceptable from regulatory standpoint. Recent advances in exosome isolation and purification techniques and the big push given by COVID to exosome-based therapeutic approaches, all this together make these days great for development of exosome-based therapeutics. We have partnered with an experienced US CDMO with very relative experience, and we're focusing on upstream processing house and will be utilizing their facilities and expertise on the downstream side. These days, we're preparing a joint grant submission as well. We have secured the use of our exosomes and core technology elements throughout our IP assets. The company owns 2 patent families today, and they're covering 2 major therapeutic areas. 1, oncology indications, and 2, antiviral therapeutics. These 2 are based on common foundation, and this results in an extremely wide therapeutic exosomes platform. Within the very active field of oncology R&D, ExoProcessor today is positioned right at the intersection of several major therapeutic strategies. While there are companies targeting p53-related mechanism of action today, our approach is more general and also possesses several competitive advantages, such as being an off-the-shelf product. We'll learn from other exosome-based companies, but as I said, our exosomes are unique. The companies appearing here and many others have been attracting substantial investments starting with very early stages of their development due to huge potential, very well recognized potential of these approaches. And finally, our vision, as I have mentioned, is to become one of the broadly administered tissue-agnostic drugs, such as Keytruda. So I have mentioned the Novocure device with glioblastoma indication and its current market cap of about 13 billion. Keytruda, with its tissue-agnostic positioning, has reported sales of over 4 billion in just second quarter of this year. Now, to summarize, our team is developing a unique solution based on solid scientific foundation with attractive competitive advantages and addressing areas of major unmet need and tremendous market. The concept is now backed by strong preclinical data, and this is a great opportunity to invest still early. I'm looking forward to interacting with you through the conference platform. These are my contact details, and I will now be happy to take your questions.